Hi, good afternoon. I'm Rick Satcher, Director of Commercialization for MTEC, and we are very happy today to present another Spotlight webinar. This one focusing on Rosalind Franklin University of Medicine and Science. And we've got a large contingent of staff and researchers on board to help explain their research and interesting collaborations. So we're very pleased to have them join us today. RFU is located in North Chicago, next door to the Captain James Lovell Federal Healthcare Center and the Great Lakes Navy Medical Centers and Hospitals. They're aligned with MTEC and are ready to collaborate across a large spectrum of disease states that affect the U.S. military and their warfighters. And starting us off will be Dr. Ronald Kaplan. He's the Executive Vice President for Research, Vice Dean for Research of the Chicago Medical School, and Professor of Biochemistry and Molecular Biology. He received his BA, MS, and PhD in Biology and Biochem from New York University and advanced training as a postdoctoral fellow at the Johns Hopkins University School of Medicine. Prior to RFU, he was a tenured professor at the Department of Pharmacology and at the University of South Alabama in Mobile. So, Dr. Kaplan, please take us away.
Thank you, Rick, for the opportunity to spotlight Rosalind Franklin University and its amazing scientists. I would like to begin by providing some background on who we are. RFU's history dates back to both well-known Chicago institutions. The university was renamed in 2004 to honor Dr. Rosalind Franklin's groundbreaking contributions to modern biology by the identification of the structure of DNA. RFU places unique emphasis on interprofessional training, practice, and the discovery of knowledge. It is comprised of 6 postgraduate health professions schools, which include the Chicago Medical School, the College of Health Professions, the College of Pharmacy, the William M. Scholl College of Podiatric Medicine, School of Graduate and Postdoctoral Studies, and a new College of Nursing. In total, the university provides graduate level education to more than 2,100 students. The university is located in the city of North Chicago, approximately 35 miles north of the city of Chicago. RFU has many exciting initiatives in biomedical technology and commercialization with the goal of accelerating the discovery and development of new diagnostics, devices, and therapeutics to improve the quality of patients' lives. Among these initiatives includes our new Innovation and Research Park and Helix 51 Incubator, the only ones of their kind in Lake County, Illinois, where 122 bioscience companies and 33,000 bioscience jobs reside, making it the largest biomedical cluster in the Midwest. We are located next door to the combined VA and Navy Medical Center and Hospital. Research at RFU is organized around centers of excellence in discovery and translational science. There are 9 such centers designed to nurture collaboration among scientists and clinicians and facilitate relationships with potential industry partners. RFU's new 100,000 square foot Innovation and Research Park opened in January 2020. The state-of-the-art facility is designed to accelerate clinically relevant research advances across complementary fields, including brain diseases such as stress and affective disorders, neurodegenerative diseases, brain function and repair, cancer immunology and infection, genetic diseases, and proteomics. The Innovation and Research Park will facilitate growth opportunities with industry, biotech firms, and startups, some of which are housed here within our Helix 51 incubator. Today, we're going to hear from several of our researchers who provide an overview of selected relevant research areas to discuss potential collaborations. As you will learn, RFU has a number of exciting initiatives in biomedical technology and commercialization with the goal of accelerating improvement of the quality of patients' lives. In this event, we are paying special attention to the needs of our warfighters. Please feel free to reach out to myself or any of the researchers so that we can begin new partnerships. Thank you for your interest in Rosalind Franklin University. And now it's my pleasure to introduce Dr. Connie Cleary.
Good afternoon, everyone. I'm so thankful to have this opportunity to speak to you about Rosalind Franklin. We will begin with an overview of RFU, followed by the research strengths and capabilities, and then we'll have each of our speakers speak about their areas of expertise. So as Dr. Kaplan mentioned, we are proud of our rich heritage. We have the 6 graduate health profession schools, and they're really at the intersection of clinical practice and exciting medical research. He also briefly mentioned that our researchers organized into therapeutic centers of excellence. And today you will hear from several of our researchers who are part of the Center for Neurodegenerative Disease, the Center for Resilience in Psychiatric Disorders, and also we also have a Center for Genetic Disease, Proteomics, and Molecular Therapeutics, where we specialize in creating new biomarkers, and a Center for Cancer Cell Biology, Immunology, and Infection. Other speakers today will be coming from the Lower Extremity Ambulatory Research Center and the Resuscitation Institute. In the Stem Cell and Regenerative Medicine Center, they examine wound healing, aging, and neurological injury. So what are our clinical partnerships like? As Dr. Kaplan briefly mentioned, we have a clinical partnership with the Lovell Healthcare Center, which includes the VA and Great Lakes Naval Hospital, and with Advocate Lutheran General Hospital, which is the 11th largest in the US. Here we could conduct clinical trials and have access to patient samples. This is a photo of the Innovation and Research Park, which Dr. Kaplan briefly alluded to. Our interest is to accelerate clinically relevant research and to facilitate growth opportunities. One of the goals is to create an environment where industry and academic researchers are easily able to collaborate. And how do we do that? Well, we do that by co-locating the office and laboratory spaces in close proximity to each other. So those collisions can occur frequently and can lead to interesting discussions and potential collaborations. We offer a range of engagement opportunities, including sponsored research, strategic partnerships, consulting, licensing, and access to our incubator. To drill down, RFU enables new opportunities through its centers and research cores, supports your pipeline with in vivo animal testing facilities and a simulation center. It's located next door to the VA and the Naval Hospital, where clinical trials are performed. And of course, we have this incubator and accelerator with the ability to co-locate with other companies and academics. Our goals are to work collaboratively with industry to enable technologies and further to license our technologies with the purpose of improving and saving lives. Today, we're going to hear from several of our researchers who will provide an overview of potential collaborations. As you will learn, RFU has many exciting initiatives in biomedical technology and commercialization, and it has discovery and development of new diagnostics, devices, and therapeutics to improve the quality of patients' lives. And now we will hear from our distinguished speakers. To begin with, we will hear from Dr. Dabrowska, an associate professor in the Center for Neurobiology of Stress Resilience and Psychiatric Disorders. She specializes in early identification and management of PTSD. Welcome, Dr. Dabrowska.
Thank you very much, Connie. Let me share my screen very quickly. All right. Good afternoon, everybody. My name is Joanna Dabrowska, and I represent the Center for the Neurobiology of Stress Resilience and Psychiatric Disorders. It's my pleasure to be here today. So our center vision is to enhance the understanding of mental illness through research that contributes to improved prevention and treatment of psychiatric disorders. So research in the center focuses on neurobiology of psychiatric disorders such as post-traumatic stress disorder, PTSD, anxiety, autism, depression. And we are very interested in really understanding the neurobiological mechanisms between those disorders and also stress resilience, and also understanding the mechanism in which we can combat those stress-induced psychiatric disorders. So we have a vast array of research capabilities in the center to examine mRNA and protein expression related to stress markers. We also use neuronal tract tracing studies to identify and investigate the stress-induced changes in brain structure and function. We also use chemogenetic and optogenetic modulation to understand how neuronal activity changes with stress and what is the role of the key neural circuits involved in stress-induced fear and anxiety-like behaviors. So research in Dr. Gianni's Urban lab, who is also the Stress Center director, is using behavioral approaches, but also brain site-specific pharmacological perturbations to investigate sex differences in a circuitry controlling stress-related behaviors and stress resilience. And she's particularly interested in the neuromodulation by neuropeptide Y in the extended amygdala. And as you might have heard, neuropeptide Y has received significant attention as this anti-anxiety compound since starting in the 80s, it has been shown to be directly correlated with stress resilience in high-risk special forces. Recently also used as a intranasal delivery in clinical trials, it has been proposed as a pharmacotherapeutic for amelioration of symptoms related to post-traumatic stress disorder. And as this mechanism of the NPY promoting resiliency are unknown, Dr. Urban showed, for example, that by preventing the stress-induced amygdala hypertrophy, repeated NPY infusion facilitate fear extinction, but also promote social interaction. And as such, it can promote the stress resilience. Dr. Urban also demonstrated in sex differences in a way that individuals respond to these NPY injections, and as such can also reveal some of the sex differences in for example, anxiety disorders and PTSD. And as a part of the resilience pathway that she is also trying to uncover, she identified the NPY responsive neurons in the extended amygdala and also some differences between sexes, which can shed some light on how and why the sexes respond differently to the NPY infusion. Research in Dr. Emil Rosenkrantz' lab, who is also Dr. Rosenkrantz is also a director of the Brain Science Institute at RFU, is using behavioral approaches, but also a variety of electrophysiology techniques and fiber photometry to determine how chronic stress, but also, for example, peripheral inflammation, modulate amygdala activity, and as it relates to mood and anxiety-like behaviors. For example, in a panel on the left, what it has shown was that prefrontal cortex can regulate emotion by sending specific projections to the basolateral nucleus of the amygdala, and by reducing the firing in the basolateral of the amygdala, basically reducing its excitability and reducing anxiety-like behavior, permitting the emotional regulation. So he has shown this effect in adult rats. What was really interesting is that in adolescence, this regulation was missing, but also this inhibitory influence of the prefrontal cortex over the amygdala was missing. So kind of a shedding light of how the adult versus adolescents can have different capabilities to regulate emotion. On the right panel, he has shown how amygdala activity can also be modulated by inflammation and, for example, peripheral inflammatory responses. So he has shown that inflammation increases the T cell content, but also cytokines in the amygdala, can cause microglia activation in the amygdala and cause BLA, basolateral amygdala excitability, increased excitability. That is also related to increased anxiety-like behaviors. And this is something that mimics also the effects of chronic stress on these anxiety-like behaviors in rats. Dr. Eugene Dimitrov lab focuses on understanding neurobiological mechanism and processing that is leading to intensification of chronic pain, but also aversive memories, which are associated with chronic pain, and general mechanisms of pain chronification. He is particularly interested in uncoupling the sensory component from the affective component of pain. So the idea is to feel the pain without the emotional distress and misery. And he is also studying and very interesting sex differences in the pain phenotypes. Research in my lab is focusing on neurobiological underpinnings of PTSD, post-traumatic stress disorders. As you probably know, the core symptoms of PTSD in humans include hypervigilance, inability to properly discriminate between stimuli that predict threat versus stimuli that predict safety. This can also involve fear generalization and disproportionately higher fear activity to threats which are delivered in an unpredictable fashion. We also see deficits in the ability to extinguish learned fear, which are one of the core common symptoms. So what we are trying to do in the lab, we're trying to use and investigate some of those core symptoms of the PTSD to use them in some of our behavioral models, including the rat models. So for example, we use a fear potentiated startle, FPS for short, which has been effectively used in rodents, monkeys, and humans, and has a really strong translational validity for stress-induced psychiatric disorders, including PTSD. And in the FPS, an acoustic startle reflex is significantly potentiated by a cue that has been previously paired with the food shock. Basically, what we then measure during the fear recall, we measure potentiation of the startle response measured during the cue or in the absence of the cue, condition cue. The startle reflex is very rapid. It occurs within 100 milliseconds. So it really gives us a very nice measure to be able to measure specifically the potentiation of the startle during in between the cue presentations. What is interesting is that the startle observed in between the cue presentation does not occur until after the cue is being presented, and therefore can be used as a proxy of cued fear generalization. So this is very important because we also know that cued fear generalization is one of the hallmarks of PTSD. What we have also shown was that this non-cued fear, so this generalized fear, is much harder to extinguish in comparison to cued fear, which also speaks to deficits of fear extinction observed in PTSD. What we have also come up with in the lab is something called discrimination index, DI for short, which is basically a percentage change of cued fear divided by the percentage change of non-cued fear, which is a proxy of how well an animal discriminates between the presence and the absence of the cue. So we have a patent application submitted for this discrimination index in which we propose that it can be used to measure PTSD's acceptability, but also help with PTSD diagnosis and help to progress, to basically have an assessment of disease and treatment progression. And I won't go into much of the details here because of, we have limited time, but just want to briefly tell you that we are very interested in peptidergic modulation of fear circuitry. And one of the research findings that we were able to show was that for example, oxytocin receptors, so the neuropeptide oxytocin receptors in the BNST, bed nucleus of the stria terminalis, we have shown that those receptors are necessary to facilitate fear responses to predictable and discrete threats, whereas they're able to reduce fear responses to ambiguous, unpredictable type of threats. So it really shows this very interesting bidirectional modulation of cued versus non-cued fear responses by some of those peptide that can be used to promote the stress resilience. And we were also able, using electrophysiology, to show some of the mechanisms of those oxytocin receptors action in which they inhibit specific projection neurons, for example, projecting to the central nucleus of the amygdala. And just lastly, I want to very briefly show you that recently we have also come up with a behavioral model of exaggerated fear response to unpredictable threats called anxiety potentiated startle. So in this model, we use an unpredictable fear conditioning design, such as food shocks and condition cues are mostly unpaired. And what you can appreciate in this model is in which you can compare the paired protocol with unpaired, such as unpredictable protocol. You can appreciate that this protocol significantly increases the non-acute fear, so then fear generalization, and this fear generalization increases with repeated testing. So also in preventing this fear extinction. And in addition to potentiation of the generalized fear, we also see a significant increase in contextual fear in this unpredictable fear protocol. So all of these protocols really give us a unique opportunities to better model some of the core symptoms of PTSD, but also gives us opportunities to test potential pharmacotherapeutics that can alleviate those core PTSD symptoms. So with that, I would like to thank you very much for your attention. And it's my pleasure to hand the presentation to Dr. Bev Statzman, professor and the director for the Center of Neurodegenerative Diseases and Therapeutics at Brain Science Institute at RFU. Thank you very much.
Thank you, Joanna, and I will share my screen. So let me know if you are not seeing the presentation mode. I will try and cover quite a bit of ground quickly. As mentioned, I'm the director for the Center for Neurodegenerative Disease and Therapeutics. And within this center, we have 5 outstanding researchers that study multiple aspects of neurodegenerative disease, as well as brain injury and novel repair and therapeutics mechanisms. And I think each of us have some potential for collaboration or at least enhancement of mutually interesting research areas. I'm going to focus on our work in traumatic brain injury, as I feel this is probably the closest link to military interests as well. But I will touch on each of our areas of expertise in the hopes of any potential interest or engagement. Please just follow up. Our newest faculty member is Dr. Holly Hunsberger. She comes to us from Columbia University. Her area of expertise is in cognitive aging and studying sex differences, as well as the role of stress and Alzheimer's disease. And her toolkit really focuses on a sophisticated array of behavioral assays to test multiple aspects of memory and discriminating differences as a function of sex and aging. And she also has an interesting component of her research having to do with sleep and wakefulness and consciousness, which again might be of interest to many of you. Next, I want to introduce Dr. Robert Marr. He's been studying traumatic brain injury for several years now and has long roots in Alzheimer's disease from a molecular and genetic standpoint. His technical toolkit really focuses on gene therapy approaches, which again, I think might be of great interest for people in this audience. And he's got very sophisticated expertise in viral vector gene delivery mechanisms, so ways to get therapeutic genes or disease modifying genes into specific cells of interest in order to fix or compensate for lost functions. And he also comes with a lot of stem cell biology, and he and I collaborate quite a bit on deriving human neurons from iPSCs, or inducible pluripotent stem cells, obtained from human patients. And this is strengthened by the addition of Dr. Dan Peterson. So he's the director for the Stem Cell and Regenerative Medicine Center. He also has been studying traumatic brain injury as well as Alzheimer's and aging. So he's been studying aging for actually many, many years and is world renowned for some of his studies there. And he's got some fantastic tools for local cellular reprogramming for repair. And I'll go into this in a bit more detail. And he also has incredibly sophisticated microscopy, as well as artificial intelligence and machine learning tools that he applies to his studies in brain injury and Alzheimer's disease. Judy Patashkin is probably the most card-carrying translational person we have in our center. So she works with human biosamples, so largely from blood, to generate gene level and RNA level biomarkers for a wide range of neurodegenerative diseases, Alzheimer's, Parkinson's. This could very readily apply to TBI patients as well, and has developed some novel bioinformatics tools to extract changes in gene patterns that can either predict or indicate a disease state. And then lastly, I'll talk a bit about myself. I have a few slides. I've been studying Alzheimer's for many years, and traumatic brain injuries, say in the last 5 to 8 years or so, and have made some significant inroads, I think, and have generated Department of Defense funding and NIH funding for these studies. I largely study neurophysiological approaches and like live cell and synaptic encoding in order to understand how learning and memory mechanisms are altered. Okay, so with that, I'll go into a little bit more detail just to give you a touch of what a standard or typical type of study is within each of these labs. Dr. Morris uses gene therapy approaches. So if you're not familiar with that is, it's a way to modify existing genes or fragments of genes, and then insert those desirable gene modifications into a viral vector, into sort of a vehicle that can bring that into the cell or neuron of choice. And these can be injected into human patients. It's actually being used now in clinical trials and in therapeutic strategies. We test this often in mouse models. And so you can inject these either peripherally or directly into the area of choice. And then it will modify that local genetic environment and ideally fix or at least improve outcomes. So here he's showing in an Alzheimer's and a brain injury mouse model using both a cognitive or memory task as well as a motor task, that in the injured control condition, we have impaired memory performance and impaired motor performance. But after his gene therapy approaches where he's reducing or taking out a gene called neprilysin, neprilysin helps to generate additional proteolytic fragments that are undesirable. You see this enhanced or improvement in both the memory tasks and the motor tasks. So that's just an applied example of the type of work he excels in, everything from identifying which gene, packaging it into the vector, delivering it to the cells of choice, and then measuring the therapeutic outcomes. And again, this can be generalized to a host of different diseases and conditions. Extending from this is Dan Peterson. So his work really is focused around repair and harnessing the power of stem cell biology to generate new cells of interest in a way to repair injured tissue or injured cells. And you can do this in a variety of different ways. His most recent and innovative approach is to take endogenous stem cells. So these are meaning cells within the brain that are normally very modifiable. And then by injecting or inserting these viral vectors that can alter the genes of these very modifiable cells called oligodendrocyte progenitor cells in the brain, you can actually convert normally inactive, non-electrically excitable cells into neurons. And then these neurons, or you can actually convert it into basically any cell you want. Here, he's converting it into neurons. And he can identify them using his sophisticated microscopy techniques, and then also show that these newly made neurons in areas of the brain that have previously been injured or say in Alzheimer's disease are impaired and affecting memory, they're anatomically making active synapses. And then with the type of tools we use in my lab, we can then record electrophysiologically that they are electrically active, they generate action potentials. And then these little squiggles down here are actually indicating that there's active synapses. So all of these squiggles are representing postsynaptic responses to glutamate. And we can show that it's pharmacologically a glutamate receptor. So taking inert cells in the brain, converting them into neurons that integrate into a network, and whether it's a motor network, a cognitive network, a sensory network, and as a way to improve and heal tissue that's been damaged due to injury, aging, neurodegenerative disease. Again, it's very broad what it can be applied to. And then the type of work I do is focus more on the functional interaction of these cells. So we said we've been working on traumatic brain injury for quite a while. We tend to do a lot of whole cell patch clamp record. So this allows us to measure the electrophysiological properties of cells and neurons, combined with live cell imaging to see what these neurons are doing as they're forming action potentials or encoding memories, and then study how this changes as a function of brain injury. So just as an example, in our mouse models, this graph is showing you it's an indication of synaptic strength. And here is our control responses in a non-injured mouse model. However, after these animals have been exposed to traumatic brain injury, you see this hyperexcitability. And you might think more is better, but here it's actually not. It's a hyperexcitable, sensitized neural network, and it is not optimized for actually forming or storing memories. Concurrent with this, we are seeing that in these traumatic brain injured neurons, the resting calcium levels within these cells, as well as the activity evoked calcium responses, are also enhanced. And again, this is not good here. More is not better. It's actually quite pathogenic and can contribute to a whole host of histopathological and memory and sensory motor disorders. And what we're also doing is developing novel therapeutic compounds, so small molecules, targeting this sort of calcium dyshomeostasis, this calcium dysregulation. And we can see them at work here. So we know that, say, in traumatic brain injury, hyperphosphorylated tau, so that's largely what's driving the CTE, chronic traumatic encephalopathy. This red staining is this hyperphosphorylated tau occurs with traumatic brain injury. And just with a few days treatment with one of our novel compounds, which reduces or normalizes this excess calcium, we almost entirely eliminate that hyperphosphorylated tau. We also see a very similar pattern with the A beta fragments that we don't want to see accumulating after injury. And this sort of, this is largely reduced with novel therapeutic treatments. So we've got a body of our research work that's developing and screening these therapeutic compounds. And then other steps in parallel with this are being done with Dr. Teresa Pape at the Hines VA. So she's got a population of veterans and military personnel that have been subjected to traumatic brain injury and a variety of other injuries. She's working at the gene level, where she's identified a whole host of gene level changes, these microRNAs. These data haven't been published yet, so I took out exactly which microRNAs these are, but they're coupled to key functions. So memory, cognition, synaptic plasticity, excitotoxicity, mitochondrial dysfunction. And what our collaboration is interested in, and again, this might be ripe for additional support or collaboration, is what these gene level changes are in traumatic brain injury patients that's sustained for months to years after the initial injury. What functions are they coupled to? And then what we're really good at is going in and measuring those actual physiological changes. So for the genes and the microRNAs that affect mitochondrial function, we can actually measure mitochondrial activity within neurons concurrently, say with calcium dysregulation or other components, and then test to see if these therapeutic interventions are normalizing these dysfunctions that we can measure and identify. And so by identifying the genes, develop targets, whether small molecules or even using, say, gene modification techniques such as CRISPR-Cas9 or siRNA. And again, this is in the area of expertise of Bob Marr, trying to normalize these effects. And then also using these human-induced neurons, ideally, say we can take these traumatic brain injured patients, we have their blood samples, we can convert those blood cells actually into neurons, study these neuronal deficits, and then try and correct it either at the genetic or pharmacological level. And then also study this in parallel in animal models. So those are some of the toolkits and the directions that we're excited about and feel like we've got a fair amount of expertise in, and we'll certainly be excited in sharing and working with others where we can advance therapeutics for these military patients. So again, it's part of a larger therapeutics development and screening profile we have going on in the lab. And we've collaborated in the past quite a bit with biotech companies, the Department of Defense, the VA, industry partners to try to advance therapeutics for these patients. So again, I welcome any questions. Please feel free to reach out to me for any future discussions. And on that note, I will stop sharing, and I would love to hear and introduce you to Dr. Noah Rosenblatt, who's the Assistant Dean for the Scholl College of Podiatric Medicine. So thank you very much for your attention.
Dr. Rosenblatt, we'll make sure you're off mute.
Yes, that helps.
Thank you.
So let me repeat myself. So good afternoon, everybody. I'm excited today to speak to you about the capabilities that we have within the Center for Lower Extremity Ambulatory Research, or CLEAR, here at Rosalind Franklin University. CLEAR is a research center that is dedicated to the interdisciplinary study of basically anything intended to improve the health and function of the lower extremity. And consistent with this focus on an interdisciplinary approach, our 8 faculty within the center have a wide array of expertise and training in areas including podiatric and allopathic medicine, as well as engineering disciplines such as mechanical and biomedical engineering, and biomechanics and kinesiology. Similarly, with this wide array of expertise, our research covers a broad portfolio of topics. You can see those lists, some of them listed here, and some of the topics potentially of interest to MTEC partners may include our work within the areas of biomechanics of locomotion, with an emphasis on balance and falls research. We also do work regarding human performance and injury prevention, and particularly as it relates to elite athletes and special operation forces. And also we have considerable work in clinical work in the area of wound healing, which takes place over at the Rosalind Franklin University Health Clinics, which Dr. Wu will speak to in a moment. That work with wound healing tends to be industry-sponsored clinical trials where the clinic will serve as either a site or the primary site of a multi-site clinical trial. So that's type of funding infrastructure is one which we have considerable history in working with. Not surprising, we also have history working with funding through nonprofit organizations such as the American Orthotics and Prosthetics Association, or the American Diabetes Association, and more traditional federally funded sources such as CDMRP, NIH, and the VA. We currently have 4 active CDMRP funded trials or research studies, many of which are being conducted in conjunction with some of the clinical and research staff over at the Captain James A. Lovell Federal Healthcare Center, which was spoken to earlier on in this talk. And just to give a little bit more information about that center, it's the only center of its kind that treats both active military as well as veterans through the North Chicago VA. And in part that's driven by its connection to the Great Lakes Naval Center, which is just down the street as well. And so collectively, working with the Federal Healthcare Center, the FHCC, provides us with access to an array of patient populations with different functional levels and different age ranges. We also work or collaborate with the other 2 VAs within the Chicagoland area, which are shown as green dots on that map. This is the Jesse Brown VA and the Hines VA. And it was also alluded to earlier that we have clinical partner sites throughout the Chicagoland area. Here you see those in blue, approximately a dozen sites. So collectively, again, that provides us access to a large potential patient population with varied conditions. And this work also, as you mentioned, within the FHCC, although some of the work takes place on their campus, which is next door to RFU, A lot of the data is also collected within our human performance laboratory. And the HPL is a 1,800 square foot, if I can go back, facility dedicated to the collection of human movement data and the quantitative analysis of this data. It houses a motion capture system and embedded force plates, as well as wireless surface electromyography, which collectively allow us to, utilizing models, identify, for example, loads at the joints during different movements as markers of progress in terms of rehabilitation or potential markers of future injury risk. We have a specialized treadmill with its own motion capture system that allows individuals to perform movements that can't be performed on standard treadmills. We have a material testing room where we conduct some of our mechanical testing on 3D printed prosthetics and orthotics, as well as a muscle strength testing room with a biodex dynamometer for standard assessment of muscle strength. And with that said, I would now like to pass it to Dr. Ryan Cruz, who is an associate professor with CLEAR, who will speak to you about some of the specific work we are doing within the Human Performance Laboratory.
Okay, so again, thanks for the opportunity to speak with you today. I'm just going to touch on a couple of kind of broad themes to give some perspective on ongoing work in our facility. So the first thing I'm going to touch on is just managing physical load or stressors placed on the body. And being part of the podiatry school, we certainly have a heavy emphasis on looking on stressors at the lower extremities. So what you can see in this video that's actively playing right now is both a standard video image of a foot taking out in a step, and then side by side, you can actually see the physical stresses or pressure placed on that foot throughout the duration of that step. We use this type of technology for a lot of applications. The image below may be a particular interest to some folks in the audience. This is a new technology that we're looking to develop, which is a smart insole system that is designed to adapt automatically to individuals' loading profiles in order to accommodate the best kind of loading profile for that user in a number of different applications. The next kind of theme I'm just going to touch on is falls and stability interventions. In addition to better understanding mechanistically how people fall or how they fail to maintain stability when it's challenged, we also have a good interest in looking at new interventions to actually improve responses to those challenges of their stability. So in the top image of this kind of section, you can see Dr. Rosenblatt with the amputee patient. This is one of kind of the key patient populations we work with down the lab. And then the two lower images, the image in the video, you can see folks doing some stability challenges while wearing therapeutic footwear. And so some of that stuff is done on our smart treadmill system. And the other one is done more in kind of the general space of the lab. And then the last theme I'm just going to touch on is monitoring and managing physical activity and function in the clinic and community. So we recognize that at times it's either not feasible or not appropriate necessarily to do all of our assessments in the physical restraints of our lab. So we've developed quite an expertise in mobile assessments and remote monitoring of folks for their physical capacities. So we have some images kind of just displaying conceptually some of our advanced physical activity monitors. The lower, larger image in this section is probably a little hard to see, but it's an actual map. And this is where we incorporated GPS technology in conjunction with physical activity monitoring to know not only what people are doing, but where they're conducting that physical activity. And then lastly, the standard kind of image there in this section shows an actual patient next door at the Level Federal Healthcare Center having their gait objectively assessed with body-worn sensors. And these sensors are very mobile, easily utilized in any setting, and can essentially be loaded up in a laptop bag with a laptop and used to conduct objective assessments of gait in any kind of clinic setting or really any setting. So that's a quick kind of rundown of some of the active pursuits associated with the Human Performance Lab. I'm actually next going to pass off the baton to Dr. Stephanie Wu, who, in addition to being a highly regarded clinician and researcher, is actually the dean of the Dr. William M. Scholl College of Podiatric Medicine. So I'm going to pass it off to Dr. Wu.
Thank you, Dr. Cruz. So as Dr. Rosenblatt previously mentioned, many of our clinical and translational research studies take place at the Rosalind Franklin University Health Clinics. It's a multidisciplinary clinic and houses the Shoal, Foot, and Ankle Center with over 25 treatment rooms, portable vascular equipment, X-ray, laboratory, and operating room, staff meeting rooms, offices, and educational spaces to provide interprofessional programs for both physician and student education, and also to facilitate clinical and translational research to advance forms of medical treatment and to help develop surgical techniques, medical devices, and advanced products. As an example of the applied and translational clinical trials with respect to better understand wound healing as well as wound infection, if you'll focus your attention on the top center photograph, we obtained specific wound tissue samples as well as serum for analysis. For the serum, we provide proteomic analysis, and for the wound tissue that is obtained, we perform RNA extraction followed by cDNA synthesis to create a genomic profile of the wounds that heal versus the wounds that do not heal. So if you'll take a look at the bottom right figure, that the one with the red and green rectangles, you can see the difference between the genomic profiles in terms of upregulation or downregulation of specific genes that pertain to wound healing. For those diabetic foot ulcers that ends up healing versus those that do not heal in comparison to the controls for non-diabetic wounds. In addition, we obtain the debrided tissue from wound tissue samples to undergo quantitative PCR, as well as 16S RNA gene amplicon sequencing to really assess the microbial diversity of intact skin and those of wounds, to really help us assess how medical conditions such as, say, diabetes or certain nutritional deficits may affect the pathogenesis of wounds of the intact skin to help us better understand wound infection. If you have any questions or would like more details, feel free to reach out. And with this, I would like to introduce Dr. Raul Gasmori, who directs the Resuscitation Institute at RFU. He is also appointed the Section Chief of Critical Care Medicine and ICU Director at the Captain James A. Lovell Federal Health Center.
Thank you, Dr. Wu. So let me tell you a little bit where we're coming from. You can see a picture now of the Captain James A. Lovell Federal Healthcare Care Center, which is essentially the integration of a VA hospital and a Navy hospital that happened back in 2010. Here, I am in charge of the intensive care unit, so I get a good perspective of what critical ill patients need. I'm a professor at Rosalind Franklin University, where we have a research lab that we conduct the work I'm going to describe in a minute. And with the help of Dr. Kaplan and many others, we have the LX51 incubator. So you can see in a relatively small geographic area, we have all these institutions that can work collaboratively and move forward in terms of developing science and having the opportunity to commercialize and develop other relationships in this broad area. So I'm going to tell you briefly what is that we do here at the Resuscitation Institute. First of all, let me see, there you go. So our main focus is to develop new treatments for various acute life-threatening events. And these include cardiac arrest, hemorrhagic shock, traumatic brain injury, and septic shock. And we have been using over the years rat models as well as swine models. We believe swine models are very well suited when we look at how do we translate this finding into humans. So most of our work has that in common, that we're looking at therapies that can be translated to patients. We also developed several years back the capability to look at mechanism by examining what happened at the cell level and organelle. Our main interest was to understand what happened with mitochondria at the time of these life-threatening events, and especially what is known as reperfusion injury. At the core of our laboratory or research institute is my role as director, the presence of Jijabai Radhakrishnan, who has been with me 18 years now. She's a molecular biologist. She's a laboratory supervisor and is responsible for the day-to-day operation of the lab. And very important is Alvin Bation, who is a bioengineer technician and research assistant who is responsible for all our equipment. And we have developed a platform for developing our own software using LabVIEW. We have our own data acquisition system, and more recently have developed various closed loop systems that enable us to deliver therapies acutely in an automated fashion. Now, I always like to give credit to my mentor, Professor Max Hariwal, who was one of the pioneers of critical care medicine in the world. He was the chair of medicine at our institution, the Chicago Medical School, and my mentor until 1994 when he left for California. So that was Dr. Weil, and he inspired me to do research in this area of cardiac arrest, and subsequently I expanded in the other topics of that life-threatening events, including hemorrhagic shock and traumatic brain injury. Through the years, we have been supported by several institutions, including NIH, the VA Merit Review System, and the Department of Defense for the last 10 years. And we have been very well supported by Rosalind Franklin University of Medicine and Science. The university denominated us as a resuscitation institute back in 2010 with a lot of support. And our environment also enables students from the Chicago Medical School as well as from DePaul University to come and participate on our ongoing research projects. Many of these ideas often end in a nice paper. So back in 2010, we started a company, Resuscitation Therapeutics, with the main purpose of to facilitate the translation of these findings into an area in which they can benefit patients. So let me tell you a little bit about some of the areas of research. One, as I said before, is cardiac arrest. And we have been doing this work for many, many years now, using rodent as well as swine models of cardiac arrest. And the model that we have used is the induction of ventricular fibrillation and resuscitation using various techniques, mostly closed chest resuscitation that mirrors what happens normally in patients in a hospital or pre-hospital environment. We have also looked at the use of extracorporeal circulation when we want to understand better what is happening during an episode of cardiac arrest and resuscitation, because extracorporeal circulation enables us to use an open chest model and access the heart to understand what is happening at that time at that level. Our work has been mostly focused on pharmacological interventions, with the idea that the heart during cardiac arrest become very ischemic. And as we do CPR, there is what's called reperfusion, which is absolutely necessary to bring the heart back. But at the same time, there's a penalty because reperfusion causes injury. And that injury is called reperfusion injury. And there are many pathways that are activated that cause reperfusion injury. So very early on, we identify this as a very important target, and our work support the concept that if we target reperfusion injury, we protect the heart, we can resuscitate with greater ease, and the heart that is resuscitated will do much better than a heart that has to deal with reperfusion injury. Some of these interventions, we are now looking at the option to conduct clinical trials so we could translate these into interventions that benefit patients. More recently, and when I say recently, I'm talking about probably 8 to 10 years ago, we started looking at hemorrhagic shock with support from the Department of Defense. And initially, what we wanted to do is to extrapolate treatment that we have found useful for cardiac resuscitation, see whether they could also benefit resuscitation from hemorrhagic shock. And indeed, we look at erythropoietin as one potential agent. And in the process, we developed a swine model of hemorrhagic shock. We did not see any clinically relevant effect of epinephrine, of erythropoietin, but in the process, we learned that administration of vasopressin as an infusion intraosseously was highly effective to achieve early, to avoid death from hemorrhagic shock. And I'm going to show you at the very end of this presentation, a series of experiments that we conducted in this model that support the idea that vasopressin infusion early in an individual who is suffering from severe hemorrhagic shock could be life-saving. This work has been done only in our swine model, and we developed two different types of model. One, which is called control bleeding, in which we remove blood through the right atrium, and we have developed a computer algorithm to develop in a way in which it mimics what occurs during bleeding, meaning that there is a very fast blood loss at the beginning and then progressively slows down. In fact, it follows a mono-exponential decay curve. And the other model, which is the uncontrolled bleeding that we developed by using liver laceration. And this is important because in the battlefield, bleeding will occur in the context of traumatic injury. So we have done work with those two models. And as I said before, using vasopressin, we have been able to show a significant improvement in survival. And I'm going to comment a little bit more in a few minutes. More recently, we developed a model of traumatic brain injury, which we added to our model of hemorrhagic shock. And the idea was that a soldier might suffer at the same time hemorrhagic shock and traumatic brain injury. And the tactical combat casualty care guidelines recommend for hemorrhagic shock restrictive fluid resuscitation to avoid what's called the acute traumatic coagulopathy that can get worse when we give fluids. So in that setting, maintaining a lower blood pressure is beneficial, but for a patient who has suffered traumatic brain injury, that might be detrimental because a low blood pressure might compromise further the brain that is suffering edema. So we wanted to develop an approach that would deal with both insult at the same time. And we thought that vasopressin could be the way of approaching this conflict of having to resuscitate in that way. In addition to that, more recently, we have become interested in septic shock. As I said before, I work at in an intensive care unit, and we face the challenge of stabilizing a patient who is suffering septic shock. And we have developed a concept that the management should emphasize the reversal of organ hypoperfusion as opposed to achieve a predefined blood pressure target, which is what usually happens in reality with the use of vasopressors. And the escalation of vasopressors tend to be detrimental and associated with increased morbidity and mortality. So we want to come up with a different approach in which the primary endpoint will be to reverse organ hypoperfusion and allow a blood pressure to be at the lower level. And we are in the process to develop a model in an automation for assessing and guiding fluid resuscitation in septic shock. We also have a little bit of very basic science research, which has to do with the finding we found several years back. We're looking at very interesting cyclophilin D, which is a nuclear protein that is transported to the mitochondria, and we found that it interact with two of the three mitochondrial transcription factors. In the absence of cyclophilin, there's a decreased expression of complexes of the electron transport chain. There's a decreased oxidative phosphorylation and therefore decreased oxygen consumption. We found that that effect is reproduced in cells as well as in knockout mice. However, when that occurs, it turns out that there is an adaptive response that is similar to what happened when an individual goes to altitude, and so that there's an improved oxygen utilization efficiency with better performance. And we're looking now whether this effect can be induced pharmacologically with small molecules, that we could create a situation in which an individual will be more able to perform better using less oxygen. So that is for the future. In terms of capability, as I mentioned before, we can do very detailed assessment of cardiac and metabolic function during acute phase and survival phase of an experiment, and understand what is happening at the same time at the molecular level. And this is my last slide, and I wanted just to share with you a study that we finished recently, in which we look at the effect of vasopressin in a swine model of combined hemorrhagic shock induced by liver laceration and concomitant TBI induced by a controlled cortical impact. So for this model, what we developed was a stereotaxic crown that could attach to the skull of the pig, and attached to it, there is an impactor that we control with our computer to produce a very severe but very controlled impact into the brain through the dura after we drill a burr hole. So here we would simultaneously induce the cortical impact and lacerate the liver. And then what we observe typically is a rapid bleeding that tend to stabilize after about 30 minutes in the animals. We would very early on, within 7 minutes of induction of the insult, start the infusion of the drug that we are investigating. And we will continue that for 24 hours and observe what happened to the animals after 72 hours. So in this experiment, we were looking at different drugs that have in common that stimulate, activate the vasopressin 1A receptor. One is vasopressin, which is non-specific because it also activates a B2 receptor. This is selective 1AB1, and this is telepressin that has the advantage that can be given intermittently as opposed to vasopressin that need to be given continuous. However, we found that vasopressin was the winner, and in terms of survival, 80% of the animals that were subject to this trauma were alive and doing relatively well after 72 hours compared to the control group that was treated according to current management of this insult. Only 33% of these animals were alive after 72 hours. So in summary, this is what we do. We're very interested in dealing with life-threatening event and finding therapies that could be translated to humans, either in the battlefield or in civilian situations, mostly in the pre-hospital arena. And I think now I'm going to give it the presentation to Connie, I believe so. Thank you.
Well, I'm happy to jump in, Connie. We're past the hour, but a lot of folks, oh, there we go. That's excellent. Great contact information for everybody. Before we do leave, yeah, we wanted to send a poll out. If you'd like any more information or be contacted by RFU, please indicate so, and we will connect you all. But so we have a few questions that came about throughout the presentation. One of them I think is addressed to Dr. Stutzman, and it has to do with the drop-seq or similar techniques on the brain tissue of subjects that have recently received stem cell therapy. Have you looked at the characteristics of these neurons to understand what might be happening?
Okay, so thank you. I had actually replied directly to him.
You did, great.
But I'll kind of expand on my answer so everyone can understand. So in terms of this direct transformation approach for like local stem cells to be converted into neurons, that hasn't yet been done in the human brain. What we are doing with Dr. Peterson is using animal models, mouse models, but using human oligodendrocyte progenitor cells inserting those into the brains of these mouse models and then converting those into human neurons, but within the mouse brain context, and then doing, a deep sequencing analysis of those neurons. So these, like I would be doing the electrophysiology, calcium imaging. Dan will be doing the morphology, and then also either single RNA sequencing to get their, their gene and genetic or their RNA signature, and a path analysis done on those. So those are ongoing studies, but those are exactly the sorts of questions we're trying to answer.
Okay, thank you. So another question around reconnecting the neurological network without pharmaceuticals or stem cells or any other therapy like that. Are you exploring anything like that, or have you already maybe adjusted to Vipin?
Yeah, I did, but just again for everyone to hear. So those aren't the type of studies we specifically are doing. Although I know that the clinical partners are engaged in more like physical therapy, occupational therapy type of approaches. So I know we have people here working with TBI patients, certainly at the level of Federal Healthcare Institute working with TBI patients. My collaborator, Teresa Pape at the Heinz VA, I know a lot of her rehabilitation approaches. So it's more in the physical therapy, speech therapy, occupational therapy type of approaches. So I'm not the clinical expert, but I can direct you to them or find them for you if you'd like to know more information.
Okay, great, thank you.
Just as a follow-up on that, I can say that one of the researchers in the Clear Center who wasn't on the call today has been involved in some exergaming interventions with TBI patients. So using things like, you may be familiar with the Wii exercise board that came out several years ago, or other interventions like that, looking at primarily improving functional outcomes in these patients. But I believe they've seen some benefits in other areas outside directly of kind of physical functioning in the studies they've been doing. And so that is a bit of work that's been ongoing on that end.
That's right. That's
right. I'm curious with your collaborations with other either academicians or companies. Have you moved anything into the clinic, or are you involved in IND enabling studies or IDE enabling studies across the board for everyone?
I'll jump in and say we haven't gotten there yet. We're sort of pre-clinical, target validation and compound development, still looking for, optimal drugs, but we've got a small library of compounds that we hope to advance and move forward into exactly those directions.
Similarly, kind of some of the more physical therapy based work within Clear, we have evidence within our laboratory setting that some of the interventions have been effective in our ongoing work is translating that and trying to figure out some of the obstacles that exist in that translation and how we can get over those barriers.
So similarly for us, we have been working with pharmaceutical companies that already have, for example, anti-PTSD compounds in second and even third phase of clinical trials, but we have been also working with them, trying to collect more preclinical data using our behavioral models, rodent models of post-traumatic stress disorder.
Okay, great, thank you. The floor is open for any questions by our members.
Well, I just want to add that we have a portfolio of technologies that we are interested in collaborating with industry, other academics or foundations to get the technologies out from the lab into the real world. And our researchers are able to collaborate on these industry partnerships with their work. Even if the work is early stage work, we can help to create the next stage of development for FDA trials and so forth. So if any of you are interested in collaborations, as Rick said, please feel free to reach out. You can also reach out to Dr. Kaplan, myself, or Michael Rosen. We really appreciate the opportunity today to present to MTEC. And I'm not sure, Rick, if there are any further questions that you have in the chat.
Yeah, I don't see any further questions. So at this time, I'd like to thank all of our collaborators and participants. We appreciate hearing all the great work that you're doing, and very happy to have you as MTEC members, and hope to see more of you. We have an annual meeting coming up at the end of May. It's May 24th and 25th. You can register on our website. We also just recently produced an annual report, which was very well done by Madison Bell and our marketing group. So we hope to see you there, and again soon on other webinars. So thank you very much for your time today. Appreciate it very much.
Thank you, everyone.
Yep, thank you.
Thank you.
Thank you. Bye-bye.
And Anna, we will be sharing the slides on the membership only website, slides and the recording. So thank you. Good day.
Sounds great. Thank you so much, Rick and Madison. Appreciate it.
Take
And
care.
I think Jill was on the call, so I want to do a shout out to Jill as well.
Hey, Connie.
There she is.
There she is.
Patience. Thank you, everyone.
All right, thanks, everyone.
I think we'll get some follow-on calls, and I think our sponsors will want to know about your work. In fact, my brain was racing on the collaboration side, Connie. We'll have some things to follow through on. You know, we've got the 50